What if the secret to curing brittle bones has absolutely nothing to do with your bone cells and like everything to do with your blood vessels?
Right, which sounds completely backward when you first hear it.
It really does. I mean, think about how we currently treat bone diseases like osteoporosis, right? Today, medically speaking, we can halt the damage. We have drugs that stop bones from deteriorating further. But if you look closely at what we're doing, it's a bit like trying to fix a crumbling house by just throwing a tarp over it.
Yeah, exactly.
We can rarely, if ever, actually rebuild that bone.
Yeah.
Or restore its original strength.
Which is incredibly frustrating for clinicians and, well, patients alike, obviously. I mean, we know the body can build bone. It did it when we were growing.
Right.
But for decades, we've basically been standing outside the construction site of the human skeleton, just listening to the noise, trying to guess what the workers are doing inside.
Yeah, we couldn't see over the fence.
Exactly. We haven't had a clear view of the exact cells pulling the strings or the specific genetic blueprints they're following to lay down new matrix.
Okay, so let's unpack this. Because in today's deep dive, we're looking at a July 2026 paper published in Nature Genetics that essentially tears down that fence entirely.
It really does. It's a massive shift.
Yeah. The researchers pulled off this multiscale analysis combining single-cell mapping in mice, these massive human genetic databases, and then stress testing over 1,000 mutant mouse models. So our mission here is to finally crack the code of skeletal disease and uncover these entirely unexpected cells that are, secretly running the show.
The scale of this paper is really what makes it so formidable. But to understand why this multi-scale approach was even necessary, we have to look at the wall the field had hit.
Right, the genome-wide association studies, or GWAs.
Exactly. So for years, scientists relied on GW. They would scan the genomes of hundreds of thousands of people to find genetic patterns linked to traits like estimated bone mineral density, or EBMD.
And it worked, didn't it? I mean, to an extent.
Oh, it did. They found over 500 genetic loci associated with EBMD.
See, 500 loci sounds like a massive success. Like you have 500 targets to look at now.
You have 500 neighborhoods is the problem. Finding a genetic locus in a GWS is, it's like finding a zip code for a criminal, but you don't have the street address, the house number, or even a description of the suspect.
Oh, I see.
A locus is just a region on a chromosome. It doesn't tell you exactly which gene in that region is the effector gene-like, the specific one actively driving the biological change.
Right, because that region could contain dozens of genes or even, non-coding regions that regulate something completely different.
Precisely the problem. And even if you guess the right gene, GWS data fundamentally cannot tell you which specific cell in the body is actually reading and executing that gene's instructions.
So it's like knowing a guy named Bob committed the crime, but you don't know if Bob is a plumber or an electrician.
Exactly. If you want to develop a targeted therapy to rebuild bone, a zip code is totally useless. You need the exact address, and you need to know the exact cell you're delivering the drug to.
Which means someone actually had to go into the bone and map every single worker on the site.
Yeah.
But, I mean, getting those specific active bone cells from healthy humans is basically a non-starter, right? You can't just drill into the femurs of healthy volunteers to see what their cells are up to.
No, ethics boards tend to frown on that sort of thing.
I would imagine.
Furthermore, the specific cells driving bone turnover don't just float around in the blood. They are anchored in this highly dynamic microscopic interface called the endosteal compartment.
The endosteal compartment, what exactly is that for those who might not know?
It's the exact boundary where the hard calcified bone meets the soft, spongy bone marrow inside. It is ground zero for skeletal remodeling.
Okay, so if human volunteers are out, the researchers turned to mice, given the high genetic conservation between our species. But they didn't just look at the mouse DNA, did they? They used single-cell RNA sequencing.
Right, single-cell RNA sequencing, or cRNA-seq. This is the technological leap that made this entire study possible, because DNA is the static blueprint. Every cell in your body has the exact same DNA.
Right, your eye cells have the same DNA as your bone cells.
Exactly. But RNA represents the active work orders. By sequencing the RNA, you're seeing exactly which genes are turned on and actively being translated into proteins at that exact millisecond in time.
So bringing it back to the construction analogy, they aren't just looking at the blueprints sitting in the foreman's trailer. They walked onto the scaffolding of the endosteal compartment, pulled aside the individual workers, and recorded exactly what tools they had in their hands right then and there.
That's a perfect way to put it.
So how many cells did they actually manage to profile this way?
They isolated and sequenced 133,942 individual cells from mouse femurs.
Wow. Over 130,000.
Yeah, the computational power required just to organize that data is staggering. They essentially created a high-definition transcriptomic atlas of the endosteal compartment. And when they clustered those cells based on their active RNA profiles, they identified 34 distinct cell types operating in that tiny interface.
34 different types of workers just at the boundary of the bone and marrow. Now, I assume they found the classic bone biology players.
They did. The usual suspects were all there. The osteoblasts, which, are the cells that lay down new bone matrix.
The builders.
Right. And the osteoclasts, which are the demolition crew that breaks down old bone. They found chondrocytes, which maintain cartilage. But because they were looking at the entire RNA transcriptome, they weren't just taking roll call. They defined the exact comprehensive gene programs running inside these non-hematopoietic, meaning non-blood-forming cells.
And the numbers they pulled from those gene programs are just wild to me. They isolated 1,886 genes that define these cellular programs. And out of those, an astounding 72.9% had never been previously implicated in regulating the skeleton.
Almost 3/4 of the active biological machinery maintaining the skeleton was completely unknown to the field.
Which completely explains why we've only been able to throw a tarp over the crumbling house. We didn't even know what 73% of the tools look like.
Exactly.
But, finding new RNA activity in a mouse femur is one thing. A mouse is a mouse.
Mm-hmm.
How do we take this massive new map and prove that any of these 1,886 genes actually matter for human bone disease?
Well, that requires bridging the gap between transcriptomics and clinical reality. So the researchers took this brand new, highly detailed mouse map and systematically cross-referenced it with 2 major human genetic data sets.
Okay, to see if the genes matched up.
Exactly. They wanted to see if the genes these mouse cells were using were the same genes mutated in human patients with skeletal disorders.
So what kind of human data are we talking about here?
2 very different ends of the spectrum, actually. First, they looked at the nosology of rare monogenic skeletal disorders.
Meaning conditions caused by a single gene mutation.
Right. This is a definitive database of 528 genes known to cause 719 severe rare disorders. Things like osteogenesis imperfecta brittle bone disease.
Got it. And the second data set.
Then they looked at the other end of the spectrum, which is polygenic traits. They used data from the UK Biobank, which is a massive GWS involving hundreds of thousands of people, specifically looking at that estimated bone mineral density, or EBMD.
Okay, and here's where it gets really interesting. Because when they overlap the mouse cellular map onto this human disease data, you would completely expect the bone-building osteoblast to light up like a Christmas tree.
And they do.
Right. But the data also showed that endothelial cells, ECs, and vascular smooth muscle cells, VSMCs, were highly enriched for these skeletal disease genes. And I have to push back here for a second because endothelial cells and smooth muscle cells make up blood vessels, right?
They do, yes.
So why is the plumbing system dictating the structural integrity of the house? Aren't they just there to deliver oxygen and blood to the osteoblasts?
That has been the prevailing assumption for a century. The vasculature in bone was viewed as passive plumbing, just, inflow of nutrients, outflow of waste. But this transcriptomic data forces a complete paradigm shift. The skeleton is highly vascularized, and this overlap proves that the blood vessels are active, dominant regulators of bone biology.
But how? I mean, if they aren't the ones actually laying down the calcium, how are they controlling the disease?
Through intense cellular communication, specifically paracrine signaling. The gene programs running inside these vascular smooth muscle cells and endothelial cells didn't just code for making a tube. They included hundreds of genes highly expressed and directly linked to skeletal biological processes.
So they're sending messages.
Yes. These vascular cells secrete angiocrine factors, which are signaling proteins and growth factors that drift over to the osteoblasts and stem cells, essentially giving them complex instructions.
Wow. So the plumbing is talking to the builders, the blood vessels are acting like project managers?
They are dictating where, when, and how bone is formed. If the vascular cells carry a genetic mutation and send the wrong angiocrine signals, the osteoblasts fail to build the bone correctly, leading to massive skeletal defects.
That is just incredible.
It is. This single-cell data proved that vascular cells are newly identified critical effectors of skeletal disease.
That is a fundamental rewrite of the textbook. But as fascinating as cross-referencing databases is, it still just leaves us with correlations on a computer screen, doesn't it? Like you found a blood vessel cell expressing a gene linked to a human disease. It doesn't definitively prove that breaking that specific gene in that specific cell will actually cause the bone to fail.
And in biology, correlation is never enough. To prove these genes were true effectors controlling bone strength, they had to break the genes intentionally and measure the physical fallout.
They ran the ultimate stress test.
Literally and biologically. The researchers utilized a massive pipeline called the Origins of Bone and Cartilage Disease, or OBCD. Through this, they evaluated over 1,000 genetically modified mouse lines.
1,000?
Yes. In each line, one single gene, many of which were flagged in their single-cell map, had been completely deleted or knocked out.
So over 1,000 different genetic knockouts. And they didn't just run blood tests on these mice, right? They physically extracted the bones and put them in a machine to snap them.
They performed rigorous biomechanical testing. One of the primary methods is three-point bend testing on the mouse femurs. They lay the tiny bone horizontally across two supports, and a machine slowly applies pressure directly to the center until catastrophic failure.
It sounds medieval. But what exactly does that force curve tell a biomechanical engineer?
It reveals the distinct structural properties of the bone matrix. So they measure the yield load, which is the exact threshold of force where the bone starts to undergo plastic deformation.
Meaning it's bending but not breaking?
Meaning microfractures are forming and the bone will no longer bounce back to its original shape. Then they measure the maximum load, which is the absolute peak force the bone can withstand before snapping completely. And they measure stiffness, which tells us about the mineralization quality of the bone.
Got it.
They also paired this with vertebral compression testing, literally crushing the spinal vertebrae to test weight-bearing capacity.
Okay, so just to recap for a second here, we've got this map, we found these new genes, and now we're breaking mouse bones to test them. So the multi-million-dollar question, out of the genes they flagged as suspicious in their transcriptomic map, how many actually caused the bones to fail these mechanical stress tests when they were deleted?
The functional validation was incredibly robust. When they knocked out the genes identified by their map, they saw significant, undeniable physical defects. Let's look at the gene PLS3, for example.
PLS3, what does that do in humans?
In humans, mutations in PLS3 cause X-linked juvenile osteoporosis. It's a severe condition where young children suffer spontaneous fractures.
Oh, wow. And what happened to the PLS3 knockout mice in the three-point bend test?
Their femurs and vertebrae showed clear structural and functional failure. Their maximum load and stiffness just plummeted. Yeah. Biologically, PLS3 is involved in the actin cytoskeleton, the internal scaffolding of the cell itself. Without it, the osteoblasts can't function or respond to mechanical stress properly. The physical test perfectly validated the genetic map.
So it wasn't just a statistical blip in a GWO study anymore. It was a validated physical cause.
Exactly. And they validated genes causing the opposite problem, too. They looked at a gene called IKBKG, which in humans is linked to osteopetrosis.
Osteopetrosis, that's when the bone becomes way too dense, right?
Whoa.
Like stone. But paradoxically, it's incredibly brittle.
That's right, because the osteoclasts, the demolition crew we talked about, fail to break down old, micro-damaged bone. It's like pouring new concrete over crumbling concrete without ever laying down rebar.
Oh, that makes sense.
The bone is thick, but it's structurally compromised. And the IKBKG knockout mice perfectly mirrored this defect in the stress tests. They also validated genes like FEX, which regulates phosphate.
And what happens when you knock that one out?
When they knocked out FEX, the mice developed abnormal mineralization. The protein matrix was there, but it wouldn't calcify properly, leaving the bones soft and prone to bending.
Wow. Okay, so they successfully mapped the endosteal compartment. They found the unexpected project managers in the blood vessels, and then they crushed 1,000 mutant bones to prove these genes are the true biological load-bearing pillars of the skeleton.
That's the summary, yeah.
But there is always one final hurdle in this kind of research. You can cure a mouse all day long, but human biology has its own unique quirks. How did they prove these exact cellular mechanisms translate to a human being?
By doing the hardest part of the study, they had to prove the human endosteal compartment operates with the exact same cellular players and genetic programs. So they performed another massive round of single-cell RNA sequencing. But this time, they isolated 125,063 cells from adult human femoral bone.
Wait, how do you even get live, intact RNA from a human femur? The decalcification process alone usually destroys the cells you're trying to study, doesn't it?
It is an absolute logistical and technical nightmare. It requires rapidly processing surgical samples before the RNA degrades, using advanced enzymatic digestion to free the cells from the mineralized matrix without killing them.
That sounds incredibly tedious.
It is, but they managed to sequence over 125,000 human cells. And the data alignment was just breathtaking.
Did the human map match the mouse map?
Human bone contained nearly identical populations. They found the human osteoblasts, the human endothelial cells, the human vascular smooth muscle cells. They also deeply profiled human MSCs, mesenchymal stromal cells.
Okay, just to clarify for everyone listening, MSCs are the adult stem cells living in the marrow that act as the raw recruits, right?
Really?
Like they haven't chosen a job yet, but they have the potential to become bone-building osteoblasts, cartilage cells, or even fat cells, depending on the signals they receive.
That's spot on. And the presence of these MSCs in the human data was vital because they are the primary targets of those signals coming from the blood vessels. Now, if we connect this to the bigger picture, the cross-species validation is what makes this a landmark paper. Between 82% and 100% of the specific EBMD effector genes that they validated in the mouse stress tests were found in their exact cellular counterparts in the human bone.
82 to 100%. That is a near-perfect translation.
It is so rare to see that level of concordance. Take that PLS3 gene again, the one causing juvenile osteoporosis. When they queried their human transcriptomic map, PLS3 wasn't just sitting in a random cell. It was highly active in human MSCs, human osteoblasts, human smooth muscle cells, and human endothelial cells.
So it's exactly the same.
The exact same complex multi-cell network observed in the mouse is dictating bone health in you and me.
Which means all the functional data, right? The thousands of crushed bones, the 34 mapped cell types, the realization that blood vessels are controlling the builders, it all directly applies to human patients.
It absolutely does. And recognizing the monumental value of this, the researchers didn't just hoard the data. They integrated the human genetics, the mouse transcriptomics, and the physical stress test results, and published it all on an open access web platform called musculoskeletal-genomics.org.
So a researcher working on a rare bone disease anywhere in the world can just log on and search this database.
It is quite literally a biological search engine. If a clinician has a patient with a mysterious skeletal disorder, they can input the genetic anomaly, see exactly which cell type in the bone is expressing that gene, and understand the mechanism of failure.
That is incredible.
It massively accelerates the prioritization of new therapeutic targets.
Okay, let's zoom all the way out. So what does this all mean? If you're listening to this deep dive while driving to work or, hitting the gym, why should you care about single-cell RNA sequencing in endosteal compartments?
It comes down to this, I think. For the first time, scientists have definitively charted the microscopic neighborhood that keeps your bones strong. They proved that your skeleton isn't just a block of calcium managed by isolated bone cells. It is a highly integrated community where blood vessels, stem cells, and bone builders are locked in constant communication. And we finally have the specific genetic blueprints they use to do the job.
And the clinical implications of this cannot be overstated. We are moving out of the era of just managing skeletal decline. Because we finally know the exact cellular addresses and the effector genes, we can start developing drugs that stimulate the right cells at the right time.
We can actually fix the house. Exactly. We are looking at the biological mechanisms required to actually reconstruct bone architecture. This is the foundation for reversing osteoporosis rather than just slowing it down. And it provides the first real, actionable therapeutic targets for severe, rare skeletal disorders that have historically had zero cures.
We finally stopped looking at the tarp and started talking directly to the project managers on the construction site. It is a massive leap forward for anyone who will ever face a bone density issue, which, as we age, is basically all of us.
It is, but this raises an important question, though, one that really challenges how we view human aging as a whole.
Ooh.
This study definitively proved that vascular cells, the endothelial and smooth muscle cells, are deeply intertwined with our bone health, essentially acting as the signaling hub for bone construction. Now, we have always treated these as entirely separate systems in medicine. You see a cardiologist to keep your arteries clear, and an endocrinologist or orthopedist to keep your bones dense.
Right, they're completely different floors of the hospital.
But if the plumbing is fundamentally dictating the structural integrity of the house, how much of our skeletal aging, the frailty, the osteoporosis, is actually just a downstream effect of cardiovascular aging?
Oh, wow.
If those blood vessels inside the endosteal compartment get stiff or diseased over time, they stop sending the right angiocrine signals to the bone builders.
That is a wild thought to chew on. Could the secret to maintaining strong, unbreakable bones in our 70s and 80s actually start with protecting our blood vessels in our 30s and 40s? It seems like if you want to fix a crumbling house, you might just need to call a plumber.